So, good afternoon, everyone. So, it is my pleasure and honor to conduct a webinar series on synthetic biology from laboratory to industries. So, my name is Bijay Singh. I am Associate Professor in the Department of Bioscience, Indrashil University. I welcome all panelists, Dr. J.S. Yadav, he is our Provost and Director. Professor Bharti ma'am, she is the Dean of the School of Sciences. And Professor Pawandhar, he is the Professor and Head of the Synthetic Biology in Jawaharlal Nehru University, New Delhi. And our all team of the supporting team, and also all the participants from the industries, from the faculties, researcher, clinician, policymaker, and the person from the industry, and many more. So now I would request Dr. J.S. Yadav to give the opening remark of the webinar series on the synthetic biology from lab to the industry. Sir, please.
Thank you, Vijay. And I like to welcome for this. I would say now second. Can we say the second seminar on synthetic biology?
Yes, sir.
First one was offline. This is now virtual, right? So welcome to this one, and also like to thank all of you for accepting our request and be the part of this virtual conference. And of course, Dr. Bharti will talk about the IU. So IU is a very young university, and we started only 3 years back. And we have, quite a good number of very good faculty in biological sciences, chemical sciences, and also engineering. And this is, when I was a director at IICT, actually, we started work on synthetic biology. Actually, there was devoted one laboratory, and where it was obviously, it was done with the Swedish company Evalva Biotech. And I'm sure later on Murli will speak, Murli who was with us at that time. Now he started off his own. And it is a very good fertile area, but I don't know why it is not picking up the way it should have picked up by this time, I would say. But the kind of young researchers I see and the kind of enthusiasm I see, I wish all of you to great career in the synthetic biology, and I'm sure one day some translation will come from this, this kind of research. So only thing I can say that good luck, and I'm sure, I hope all of you will be able to attend all 3 days. There's not much load. It is only, I would say that less than 2 hours every day. So I hope that you'll be able to enjoy the speaker's lecture and all. So wish you all success in your career, and also wish that this conference will have a good take-home message from for all of you. And I wish good luck. Thank you very much. Dr. Bijay, it is to you now.
Thank you very much, sir, for the opening of the webinar. So now I'm very happy to brief about what we are going to do in this next 3 years. So as we know, the synthetic biology is the one of the fastest growing area where engineering principle is applied into the biology, and the synthetic biology have made tremendous progress in the area of the healthcare, diagnostic, energy, and the environment. So synthetic biology along with the metabolic engineering, genome editing, we are going to learn in the next 3 days with the number of the application that can we go into the lab to the industries and also the clinics. So in next 3 days, we are going to have our understand more about the synthetic biology, where the computation is going to involve genome editing, where how we can do the genome editing technology in the number of the organism, and the metabolic engineering for enhancing the production and yield of the for the industrial product. So this conference will help us to improve our knowledge, understanding, and human resource in the area of the synthetic biology, because we are lacking behind a lot of manpower in this area to conduct the experiment. And this webinar will give us opportunity to learn more about the synthetic biology that can apply in the industrial biotechnology area. And now at the Indrashil University, now I would like to request Professor Bharti Dave ma'am to brief about the what we are doing at the Indrashil University and what we are. Please, ma'am.
Just okay.
Yes, ma'am. This is yes.
Is it visible?
Yes, ma'am. This is perfect.
Very good afternoon to all of you. Just to introduce to you brief about our university. Of course, Dr. Sangram was here with us last time when there was a first conference on synthetic biology, but I suppose Professor Dhar was not here last time. And for all the others who do not know about our university, let me give you a brief. So this major source of inspiration for establishing this university was Shri Indravardhan Bhai Modi, who is the founder chairman of Cadila Pharmaceuticals Limited. And this Cadila Pharmaceuticals Limited was established in 1951. And now, as of now, it has its presence in 90 different countries. It has over more than about 10,000 or more employees. So Shri Indravardhan Bhai Modi was a core nationalist at heart. He firmly believed that medicine should be affordable and should reach to the last person of our society. Even though, as on today, even though he's not here, he's not in his absence also, Cadila is still working on this motive of caring for the people. So Indrashil University was established as a philanthropic initiative of our founding chancellor, Dr. Rajiv Modi, in loving memory of his parents, Shri Indravardhan Bhai Ambalal Modi, Srimati Sheelaben Modi. If you just see to the first five alphabets of Shri Indravardhan Bhai Modi and Sheelaben Modi, the name of the university has been derived from the first five alphabets. So to honor the legacy of care and compassion, the university's governing body had determined holistically to develop this university into a hallmark of global academic excellence in terms of teaching, in terms of research, in terms of innovation. If I just give you a brief idea on the success of our university, several milestones which our university has received just in 3 years. We are into our 4th year, and our university just recently has received the Best Young University of Gujarat Award in terms of teaching and research. So as I've told you, Cadila Pharmaceuticals was established in 1951. In 2016, they took up an education initiative of establishing Sri Saraswati Education Foundation here in Rajput, which is near to Kadi and which is 60 kilometers from Ahmedabad. But it was in 2018 that Indrashil University was established as one of the first life sciences university of our country. In 2019 and in 2020, because our vision is to aim even for innovation, Niti Aayog government of India has supported us with Atal Incubation Center with its flagship program on to encourage youth of Gujarat to initiate that start. Please start screen. Okay, there were some technical issues regarding connection. So we have Dr. J.S. Yadav as the Provost of our university. He's also the Director of Research of our university. He has been the former Director of IICT Hyderabad, and he has to his credit the highest level of awards which have been accredited in science by the Government of India. That is Shanti Swarup Bhatnagar Fellow, J.C. Bose Fellow. He has about 150 plus patents, about more than 1400 publications, more than 250 students have earned their PhD degree with his guidance. Still, we have around 11 to 12 students working for their PhD with him in this university, and more than 30,000 citations. You can just imagine what would be the vision of our our provost, and due to his vision, our university has progressed to this level. So Indrashil University, as I've told you, excels in academics, excels in teaching, excels in research as well as innovation. So because it is supported by Cadila Pharmaceuticals Limited, we provide enough of industrial exposure to the students. In terms of internship, we have around 7 to 8 months of credited internship program, which is mandatory, and when the students have to do when they are in their MSc semester 4. We also focus on making the students to be well-rounded. We believe in 360 development of the students, and we have tried to achieve global academic standards, which I would be telling later on. So under the umbrella of Indrashil University, we have School of Engineering, we have School of Science. School of Science, we offer BSc Honors in Chemistry, BSc Honors in Biosciences, MSc in Chemistry, Organic Chemistry, as well as Analytical Chemistry, MSc in Zoology, and MSc in Microbiology. Whereas in the School of Engineering, we offer BTech in Computer Science and Engineering, Mechanical Engineering, Chemical and Biochemical Engineering. PhD is offered under both the programs. And we have entered into international collaborations with 3 different universities. One is Sacred Heart University, USA, Tuskegee University, USA, and University of Newcastle, Australia, where the students, if they avail this program, they would get a dual degree, one from Indrashil University, the other from the foreign university. Our university, we have a very sophisticated state-of-the-art infrastructure facilities in terms of equipments too. And one of the very, you can say, high-ended equipment that we have purchased recently is NMR spectroscopy. This NMR is being made available to both our MSc students as well as PhD students. So as we are supported by Cadila Pharmaceuticals Limited, our major aim is to make the students industry ready so that when they come out of this university with a degree of this university, they are easily absorbed by the industries. So a number of academia linkages with the university, to name a few: Cadila Pharmaceuticals, Karnavati Engineering, CII Entrepreneurship Development Institute of India, which is here in Gandhinagar, VFL, Datawise, Jackson, and Recipol Adhesives, Zydus Cadila, Torrent Pharmaceuticals, etc. And we have also entered into collaboration with a number of, you can say, CSIR laboratories, one of which is Indian Institute of Chemical Technology, Hyderabad, National Chemical Laboratory, Pune, CSMCRI at Bhavnagar, Central Drug Research Institute at Lucknow, where our students, they regularly go for their internships for about 4 to 5 months at these CSIR laboratories. So as I've told you, Indrashil University provides 2 platforms to the students for their startups. One is Atal Incubation Center, supported by Government of India, Niti Aayog, and Indrashil Innovative Foundation, exclusively supported by Government of Gujarat. And in these incubation centers, we provide access to world-class R&D laboratories. We provide dedicated office and a laboratory space, about 50 lakh of rupees as kickstart support for the students, provision of 2 crore seat fund support, hand-holding by renowned mentors, which may be from industries, government departments, R&D laboratories, and 360-degree support if they want to take up startup to a sustainable business. So this is all about Indrashil University. The students can reach at this website, they can email. Thank you very much.
Ma'am, can you stop sharing? Yes, fine. So thank you so much, ma'am, for providing the idea about the Indrashil University. Now I'm delighted to introduce today's speaker, Professor Pawan Dar. He is the head department of the School of Biotechnology, Jawaharlal Nehru University, New Delhi. So he received his PhD in human genetics from BHU Varanasi, and he worked as a scientist, senior scientist, and the group leader in the Bioinformatics Institute, Singapore, Riken, Japan, and teaches Hyderabad. And he established the research laboratory in India, Japan, and the Singapore. And he is the expert member of the European Science Foundation, and he's the one of the key person who really started and picking up synthetic biology in India. Whenever he goes where, so he start more pushing the synthetic biology, and for his more key finding, it was published in the number of the research paperwork, including in the human genetics area, system biology, synthetic biology, with the computation and the experimental validation. And he developed the computational tools where the not coding DNA, it is not non-coding, it is not coding DNA, which is generally not produce the protein. And he developed this tool and experimentally validated, and he produced the protein from the not coding DNA. This is the dark side of the genome. And he was able to produce this therapeutic molecule, and it has more potency and the power as compared to the regular protein coding gene. And he is the person to initiate the one of the biggest competition in synthetic biology, that is called the International Genetic Engineering Machine Competition. So it is every year. It is held in the United States. And he is pushing this, and now the government of India, Department of Biotechnology, is supporting the Indian team, at least 5 teams, to go and join the big competition in United States. So he is the person to start such kinds of the activity. So today he will give a talk on the synthetic biology concept to principle. So I would request Professor Dhar to please share your slide and give some information what we need to know more. Thank you, sir.
Thank you very much, Vijay, for your kind introduction and invitation to this very important meeting. I'm just going to share the slides. Is the screen visible?
Yes, sir. It's perfect, sir.
All right. Thank you very much. So I'll just switch off my video so that I can stay within the bandwidth. All right. So first of all, thank you so much for arranging this very important meeting once again and taking the synthetic biology community participation forward. India has taken several strides in the past, as we all know, but still somehow we have not been able to make a big impact, probably because industry needs to adopt it and more academic programs need to also take it forward. We have done our own small bit. However, there's a scope of a lot more activities. Because there are many students today, I thought of putting together some of the conceptual slides in the beginning, introducing synthetic biology, the way I understand, and then I'll point out to some applications, and the third part of the talk will be about the work that we do in the lab. So as you all know, reductionism has been the predominant scientific approach in biology, which means that you look at an organism, say for example, Gregor Mendel looked at the pea plant, and then he was keen to know how the characters are transmitted. So he had to imagine something like factors, which he didn't see, but he could imagine it, and then conduct many experiments to validate his hypothesis. So what he was trying to do is to look at the organism and reduce it to a set of factors that would represent the organismic behavior. So the whole approach of going from the top level, that is the organism, to the level that basically decides what happens within, the whole approach is called reductionism. So when you look at this level, then you find out there are many systems which are gel together to run this, the entire organism. But when you go a little deeper, you notice then there are more details, and you call this as gross anatomy. When you go a little more further, using microscope and the staining technologies, you notice that there are cells that make up the tissue. And when you look within the cells, you find that given the right microscopic power, differential staining techniques, you notice that the nucleus that was looking very innocent, or the cytoplasm that was looking absolutely barren, now has a lot of details. Now, when you go deep into those details, people found early on that there is something called as a chromosome. So as you can see from the top level, we are going slowly to the level at which there are many components that play, and then that gives rise to a collective interaction, and the phenotype is the outcome of these interactions. So now when you look at the chromosome, you see that it's made of DNA and proteins together, and DNA coils itself 7,000 to 10,000 times to make a chromosome along with these proteins, and likewise uncoils itself. So when they found that DNA was the mega star that was doing a lot of things, so they felt that that was 40s and 50s, people felt that this is the main molecule that we need to chase. And once you understand this, you have understood life, because this molecule is transmitted. But over the time, last 5 decades, the RNA and protein community have caught up, and they have established that it's not just the DNA, but DNA gives out certain signals, and then RNA is produced, and protein molecules are made, and then you have a regulatory RNA and so on. So DNA actually lost this, the superhero status, and it is now considered more as a database of nucleic acids. This tells you how the science has evolved. And so this is one level of reductionism. When you go further, you move from these megastructures to molecules. Here you're entering the domain of chemistry. Because it happens in biological system, you call it biochemistry. Now, when you go further from molecules, you see there are atoms. Now you're at the level of fundamental physics. If you want to go further, you realize that the bulk of the atom is space. Now, you do not have any GFP to light up the space, or some staining technique to light up the space. So here is the point where you lose resolution, and you do not know what to do next. You cannot understand space the way you have understood till you reach space. So it's better to zoom out and stay at the level at which we can do our analytical techniques and the measurements. So the whole idea of understanding a system is to uncover its components. So this approach of reductionism has generated a number of genes, RNA molecules, protein molecules, and so on. But now, when the Human Genome Project started, or a little bit early to that, there is a lot of information generated, and people were worried, how do they manage this information? So then people have to use, they have to transfer the data to the databases using computers. And the field of bioinformatics was born, and now modeling had a role to play. So when we move the molecules to database, and then it's not only moving the molecules, but also their interaction patterns, the way they interact, protein-protein and protein-DNA interaction. When you move them to the computer, then you try to understand cell virtually, instead of physically, you understand the whole. So you're doing a virtual biology instead of investigating it experimentally. Now the third approach started back in, and the seeds of this were sown sometimes in 2000 or a little earlier than that, when people from Boston and Berkeley area, they came together, they discussed, could there be any engineering approach to biology? So you have two ways of doing biology. One is reductionism, the other is integration. The third approach that was proposed, and then there were 2 papers which were published in the year 2000, that gave the foundation of the first meeting of synthetic biology back in June 2004 at MIT, where they established the engineering or the construction approach in biology for the first time. So the first approach is popularly called as molecular biology or biochemistry. The second one, we call this as bioinformatics if it's a single component study, or systems biology if it's a multi-component collective behavior of the molecule. The third approach was called synthetic biology. Now, this is not to say that synthetic biology was accepted when it was proposed. I remember there were a lot of discussions and debates and lack of acceptance early on. And people who were promoting the word synthetic biology, they were asked a lot of tough questions because people thought it is something to do with chemistry. It is synthesis of something, while as this is not only synthesis, but also the standardization part, building standards and so on. So Drew Andy, who was leading this group from MIT, he was then he suggested another term, constructive biology. Well, people said if constructive biology is the way to go forward, then is the rest of the biology destructive? So that was not accepted. Then somebody said, let's call it biological technology. Well, if you call it biological technology, then what's biotechnology? Likewise, many terminologies were shuffled through. And finally, a term called biological engineering was proposed and accepted as an equivalent. So if you have publications, you see them in biological engineering journals or using the word biological engineering, or there are some publications in the journal synthetic biology. Both of them mean the same thing. It is just the preference of people. Because we have a lot of reductionism data, a lot of that is generated. So I proposed back in 2007, is this the time to construct a periodic table in biology? And if yes, what should be the element of this periodic table? Now, why we are talking of periodic table here is because when you're talking of engineering, you have to bring some certainty to this. Now, one may say that we have already done genetic engineering. So genetic engineering as a term is already popular for many decades. What is the need for another engineering? So I want to clarify that genetic engineering is actually a misnomer because we never engineered genetics. All we were doing is probability. So it's like you have thousands of vector molecules which you have constructed in the lab, and you have millions of cells growing in the Petri dish. And then you, through electroporation, usually, you transfer these vector molecules to the cells which are growing there. And you hope that some of these molecules, the vectors, will get access to the cells. And then you hope that some of them will align at the right spot within the cells. Now, out of maybe thousands and thousands of cells you grow, maybe you get 6, 10 or 8, 10 or 20 colonies. And then among them, you see that the integration has happened in the right spot in few of them. Now, this is a probability. This is not engineering. Engineering has just begun, and I'll defend that in a while. So the question is, can biologists use engineering approach to construct systems? So why are we talking of biology and engineering in the same breath is because there are many similarities between the two. And obviously there are many dissimilarities. You don't have a fixed component attached to a certain cell substrate. You have moving components, unlike the motherboard that we use so let's say if you have to design computers. Biological systems are predominantly analog and non-linear. And also the only laws that we are familiar with, if you call them as laws, that are Mendel's, and they are not useful because they are zoom in and zoom out types. They zoom in from phenotype to genotype and zoom out from genotype to phenotype. They were not designed for engineering purposes. So again, we have to begin from the scratch. And we know that our systems are noisy, and unpredictable noise means the fluctuation of the expression that you get. And we have just gone through unpredictability. When you construct a circuit in engineering systems, you have these nodes very well laid out. So when you model the whole thing, you know how the information is flowing within my system. But even to make a 2-point contact within the cell, you have to go 2 levels down. Actually, you have to go several levels down. You have to ensure that the molecule is not only made, but it is not degraded fast enough so that the next event will be affected. You have to make sure that the molecule is not only synthesized, but it also survives, and it also finds the right spot, and only then you go to switch the next event on. When we make an analogy between engineering and biology, one has to keep this in mind, that it's not that straightforward. So there will be gaps, there will be unexpected behaviors. Models will not exactly behave the same way that they behave in engineering community. So therefore, to design well-behaved systems, one needs standards and rules of composition. So I told you that genetic engineering is not engineering, it is probability, because we never cared about standards. We never needed that, actually, because the whole idea was to do reductionism. But now when you need standards and rules of composition, what are the standards we are talking about here? They are the ones which are of the level of IEEE standards of engineering community. These standards don't exist in the natural system, so we have to make those standards. Therefore, it's good to use engineering as a philosophy. Absolute engineering solutions do not exist for biological problems, because probably engineers never faced. Now, I'm not downplaying the role of engineers, their contribution. It is just that by the very nature of biological systems, there is a lot of probability. So when the signals have to move, say, for example, if you design a Boeing 747 aeroplane, you are stitching together nearly 7 million parts and hundreds of kilometers of wires. Now, isn't that a complex system? Yes, it is. But if you have to make an E. coli, it can take you decades. And still you will not be able to make it, because it is a non-linear, it is an analog system. Molecules are floating around, they're not sticking to one place. So it's much harder. There are probability events, there are concentration gradients, and so on. So what's the solution? Well, it's not a solution, it's a practical way out, that we have been good in decomposing the system one part at a time. We call it gene knockouts or mutations. But so now the next phase is to compose the system one part at a time. And we call this in the synthetic biology, this is what we do. And the whole idea is to do a controlled construction. This has also given rise to the concept of a truth table. The truth table concept was proposed in this first meeting. And people were of the opinion that if they make 4,000 truth tables in E. coli, then they can design E. coli, let's say 4,000 truth tables, meaning that 4,000 genes will be monitored for their behavior, and they can then assemble it. But this didn't happen. Initially, it was felt like that, but it was pretty tough getting this truth table is not a problem, but making use of the data is a problem, because it's not portable. Because these values are specific to the strain, specific to that particular culture condition and the metabolic state in the cell, which is changing all the time. So these values are changing all the time. They're not static. So you cannot use them to construct systems. Synthetic biology has gone through many definitions, and there are many administrative and political definitions, but this is a brief scientific definition that we feel comfortable with. Is a rational design and controlled construction of biological components. When I say components, I'm talking about parts, devices, and circuits. Parts as in genes, RNA, devices as in operon, and circuits as in metabolic pathways. As I said, biological engineering is also used as an attractive equivalent of synthetic biology. And some people like to use this terminology more than synthetic biology, because synthetic biology got a bad press. That was a little unfair, but nevertheless, people got sensitized to the fact that it can lead to many applications which are undesirable. Companies have used synthetic biology approaches. Gene Power Company, Opix Biotech, you can read it faster than I can speak, and Metabolics Company. From India, there has been participation in the international genetic engineering. This should be genetic engineering, machine computation. As you can see over time, the participation has increased, except last year because of COVID, it dropped down. And we proposed to DBT that there should be a prior to IGM competition in India. So we started something called as IBEC, which stands for Indian Biological Engineering Competition, which essentially means that the applications are sent to DBT and there's a panel. It goes through all the applications, and then people who are selected, they get up to 10 lakh rupees of funding per team, and then they go to MIT to present it. It would have been ideal if we had our own competition. I've been trying to plan that for many years, but unfortunately, we've not been successful so far. So the pressing need of the hour is really to have an Indian synthetic biology network with an Indian biological engineering competition. And if you look at, if you just do back of the envelope IGM maths, you realize that we have spent, as a country, we have spent close to 4 or 5 crores so far on the IGM competition. Imagine if only half of this money was available for us, we could have done wonders. And we don't need to go to US necessarily for this competition. We can ourselves, take care of this because we have the core competence. We just need some hand-holding and prolonged support. So if you are interested and if you have intentions of scaling it up, so please let me know, we can work together. The IGM registry of standard biological parts has more than 20,000 entries. But the question is that these parts do not work as specified. And when you mix and match them, they do not follow a certain prediction. And also the concern is, for how many cell divisions will your design be robust? Because you construct something, say you construct a wall, and then you find that it is standing there for many decades. But now if you construct a gene network, maybe a 3-gene or a 4-gene network, will it stay there or will it get disrupted after some time? So that's the problem we are grappling with. So instead of going from, constructing devices from parts, the proposal is that if we construct, if we, let's say, map the pathways to vectors, then we have eliminated all the uncertainties that we are coming across while assembling the biobrick, parts together. So these biobrick parts together, when you mix and match them, then you construct a certain circuit for a certain behavior, but the behavior will be changed over time. So now if you're focusing on only pathway, so you have a mega cloning vector and you put the entire pathway on the vector and you plug it in and plug it out, then you have a better control and much better control and faster way of delivering the product. So can we do this? We do not know really. So the first event in India was held when I was in Kerala. And then this was, we started a Sanjeevani network, and it was proposed by a PhD student, Ashwini. Then we had the second event at JNU, then the third one in 2017. And after that, I guess Indrashil has taken over, and I'm so delighted and thankful to Vijay for taking this forward. Well, coming back to our work on the dark genome, you see, traditionally by converting genes into junk and garbage in terms of knockdown and knockout, we have learned biology, right? So the question is, can we make brand new genes from the so-called junk DNA? When you say junk DNA, this term was coined by Dr. Susumu Ohno back in 1920s, and he thought that there is a lot of desert where there are no genes. So he called it junk, and then it somehow got stuck because he was a very famous geneticist. So people like to use his terminologies and feel excited. But later on, it was proven that what he felt, or what the entire community felt was junk, was actually RNA coding genes. So you have a lot of RNA regulatory elements, which are making the bulk of the genome. In other words, genome has this sort of a headache. To manage a minority, it needs a majority of rules and regulations, which is what happens in university anyway, all the time. So we are very, very genomic in our way of doing things, that we have a lot of rules and regulations for managing a a group of students. So, but there are still some sequences that do not express themselves. These are typically the junk. These are authentically the ones that they do not have any expression profile. There's no RNA coding, no protein coding property. So we said, how about this not coding, not non-coding, but not coding, a non-expressing fraction, the dark matter? Can we make something from there? This also came up from this observation that there are start and stop signals in specific spots in the genome. They're not randomly placed, they're placed very intelligently. The question is, why nature places signals at specific spots? How did she know that I had to place a start signal here and stop signal there? Because if it was random, then, or if it was tuned to a certain organism, then if we take it in information, then we can create more variants from there. Is it possible to do that or not? So that was the question. So if we have to prove our hypothesis that the non-expressing genome can be made to express into functional genes and proteins, then our choice of sample should be very, very important. So we took intergenic sequences. This is E. coli genome, and we took intergenic sequences with no transcription history. So the lines that you see in red color, the arrows left and right, as you can see, we took a number of variations, overlapping, non-overlapping, facing each, in opposite direction, small and big, because we did not know what will work. And there were many odds against this idea, as you can see. When I took this idea to my students, I was in Japan at that time. My PhD students and postdocs refused to work on this project because they felt that this is a risky business, a risky proposition. And if they fail after 2 years, they will not have any backup because, they need to work on something that leads to publications. So we hired a technician from Singapore. She was a tissue engineering expert. So we hired her and we trained her in molecular biology. And she worked in this project, and then she generated a lot of results. And then everybody came back and started contributing. And as a mark of respect for this technician, she was a co-author on the paper. So the question is, how do you make a gene out of a non-expressing or a not coding DNA, which is does not code for RNA and proteins? Well, this is pretty simple because in the textbook, you know what is a gene. How does it look like? You know this pretty well. But when you're playing at the genome sequence level, you need to care about many, many things which can prolong the whole effort of making brand new genes. You need to care about their the order and the mutations and so on. So we said, let's not even go there. Let us do a quick synthesis by pulling out the information outside the genome, which means that you take all the information using bioinformatics, you do many filters, select your sequence, PCR amplify this, and just insert in the vector. So while we were wanting to make a vector, we found that Invitrogen had released a PBAT topo around the same time. We were very thrilled because that solved a lot of our problem. It not only had the signals that we needed, but it also had epitope. Now this was a histag wherein you have these antibodies, and so which means that essentially what it means is that when your protein has this tag, you can detect it during the Western blotting. And if there's a color, is a colored band, you see that this is your protein. If it is absent, then the protein is not being synthesized. So that is exactly what we found. In absence of the inducer, there was no band. In presence of the inducer, there was band. It corresponded with the molecular weight that was calculated earlier. So now the nature of questions changed. Now the questions were, do they fold into certain polypeptides or certain structures? And what would be the structure of, is it a stable structure? What would be the structure of such kind of a synthetic polypeptide, which has never been made by the organism? We had 42 NMR machines in our campus at RIKEN. but we could not access even one of them because these were all working for the Protein 3000 project of Japan, wherein our scientists had to solve 2,500 structures in 5 years, and rest of the Japan 500 structures. So we could not access it, unfortunately. So we have to live with the computational predictions. And this was done at the Bioinformatics Institute of Singapore, where I worked earlier. And they helped me in predicting this, the structures. So the question was, what are these guys doing in the cell? We found that we had initially taken 10 samples, we lost 4 of them. And among the 6 samples that we tested on different cells, we found that one gene expression is repeatedly killing the cells. So we said, if this is right, can we restore its growth by switching off the gene? And that's exactly what we found, that after 2 hours, nearly, when the numbers have started going down, we switched off the gene, and then it went, the numbers of the cells went back up again, thereby proving a linear correlation between 2. Well, the story is not that simple because, remember, we did not design the vector. So vector contributed some amino acid sequence when it got translated. It had some overhangs at both the ends, and a lot of that actually. So that means the results that you just saw, they were not exactly coming from the not coding DNA or the non-expressing DNA. They came from the non-expressing plus vector contribution. So we said, let's do something about it. We changed the entire strategy, used a different vector, and took a sequence wherein half was the vector contribution and half was coming from this intergenic sequence of E. coli. And earlier it was not affecting the cell, but then when we changed the strategy, we could remove the vector contamination. And then we found that there's something interesting. So the functional study of the lab-made genes, as we call them, can be done if you overexpress them. You see, currently we do not have a knockout library of the non-expressed genome. We have the library only of those protein coding sequences that we know about, which have been characterized. But if you remove a not coding sequence, will it affect the cell? We do not know. The Encode project has very clearly established that the bulk is a transcriptome, a tiny part is proteome, and so on. All we are asking is how many non-expressing sequences can be encoded into functional genes and proteins. And so we have done this. So then in the second phase of this project, it moved from, because remember, first was, it was our good luck that we got this phenotype, but we didn't want to end up again in such a situation. So we said that let's make a computational database and put a lot of filters and say that if I model every intergenic sequence in E. coli, Drosophila, yeast, and now humans, that's what we're doing, what kind of proteins would be made in terms of structure, in terms of function, their address, their interaction, and so on, in terms of the protein disorder as well. So that gave us many predictions, and then we found very interesting observations, and we totally focused on therapeutic application. We did not go to the enzyme and other levels. We focused on therapeutic applications. So, for example, a Drosophila melanogaster intergenic sequence, when we computationally tested, it was showing amazing overlap with the alpha defensin protein, which is a peptide which is marketed as an antimicrobial peptide. Sequences were totally different and structures were nearly same. And when we synthesized this peptide and tested, we found that we are getting good results. Okay. Likewise, we checked with anti-malarial peptides against these 3 targets, and we found that in presence of these peptides, which were derived from the intergenic sequence of Saccharomyces cerevisiae, we found that a bulk of these parasites are unable to enter RBCs in presence of these peptides. This was another totally crazy idea. We said, how about tRNA peptide? Because tRNA has been doing a social outreach for millions of years. It has been ferrying the amino acid to ribosome and helping the cell to make polypeptides. But it never got a chance to express its own sequence into an amino acid sequence equivalent. So we said, how about doing that? Now we know that there are steric hindrances and so on, we cannot push the tRNA into ribosome structure. So we said, let us assume that these peptides can be made, and let us do a bioinformatics. And then from there, we chose certain sequences, a lot of filters, and we chemically synthesized these. And there was an amazing result that this tRNA encoded peptide, or Trep, what we call them, we found that, and we tested them on malarial parasites, leishmania, and many microbes and so on, because we don't know what's going to happen. And we found that they are showing amazing correlation, IC50 value and 30 nanomole. We see that this is the kind of picture. And as you can clearly see that it kills leishmania, but not only this, when you increase the peptide concentration even 10 times, 20 times, human cells are not affected. So that was the best part of it. And now we are making a lot of them. And likewise, we found the other molecules which are in the process of validation. And all of them are coming from, so to say, junk DNA or non-express genome, or what we like to call sometimes the dark matter of the genome. Well, this led to many invited talks, and I had a fortune to publish the genome engineering book with Vijay, who has been doing bulk of the work. So not my contribution, he's the sole contributor of this book. And I'm so happy that he's carrying forward this mission. So this work led to spinning off the company at JNU. And so in future, we are wanting to make a lot of them and then work with the industry. We have some inventory. The best part is that you own the IP of every molecule that you make, because you find that nature did not make it, at least in comparison to the data that we have seen so far. So since everything starts from junk, sometimes my students like to call this new area as junkomics. On an outreach aspect, we have been, well, we started this Systems in Synthetic Biology journal. This was with Ron Weiss, who was at that time in Princeton, then moved to MIT. We ran it for 8 years and we closed the journal because a lot of new journals and Nature and other journals, ACS journals, they started getting into synthetic biology, and it was difficult for us to manage the show, so we had to stop it. But we initially ran it at the time when there was no journal. This was right after the field was announced. And we have also been training students. We have just completed this 3-year training program. And we have submitted a report to DBT on the policy aspects of synthetic biology. How do you regulate it? What are the various do's and don'ts? So it has been 1-year project with Balakrishna Pisupati. So Dr. Balakrishna Pisupati, he's on the panel of the United Nations for synthetic biology. So he was greatly helpful in advising and shaping up this whole work. We have recently joined a global biofoundry network, which is run from UK. And we are hoping that synthetic biology work that we are doing, this will become eventually part of the biofoundry. And this work has just started, so not much available. These are the people who are involved in the work, and we received funding from various sources, and we are so grateful for all the support. And there has been a challenge always, because when the whole work started, people downplayed this and discouraged us from moving forward, but we somehow still persisted, only with one idea. For the last 10 years, we are just doing one project. We're not doing 10 projects, just one project. And so, this comes from this small atom, which has a big motivational message for us. Be positive, even if you're always surrounded by negative things. So thank you very much for your kind attention. I realized that it's time for me to stop, and we will take the questions later on. And I'm personally very keen to listen to my friend, Dr. Sangram, who is here today. Thank you very much.
Thank you so much, sir, for the very informative junk is not really junk. It is very, very informative. And so the tremendous translational things that we will see a lot of futures there. So now I would like to introduce the second speaker, Dr. Sangram Bagh, sir. He is associate professor in the Saha Institute of Nuclear Physics. So he completed his PhD from the University of Toronto, Canada, and the postdoc from MIT. Then he is the key person to work on the cellular computing and the synthetic biology. And he is member of the number of the peer-reviewed journals, and he is also the fellow of the Ramalinga Fellow and received number of awards. So without spending a lot of time, so we are very happy to listen in. Sir, for your information, as we have participants not only from India, we have participants from Malaysia, Vietnam, UK, and South Korea. So I'm so happy and glad. So people is listening you and getting some of the information. So Sangram sir, please.
Okay. Thank you very much, Professor Singh, for such a nice introduction. And also, I mean, it is a great pleasure that you invited us, actually, and it's an honor to participate in this meeting. And it's another pleasure is definitely to hear Professor Dhar. I mean, we are not seeing for a long time physically, but still this gives a good opportunity. And I think, as you heard Professor Dhar, that he covered the fundamental ideas of the synthetic biology. And I also, so when I came to the India, so he's the key person who actually invited me and just tried to tell the other people that this is another guy who is trying to do the synthetic biology. And I also know that's what Professor Dhar said, that's the negativity. So, I mean, when the Indo-US conference, when the Professor Dhar was actually organized, that time I heard a mixed repercussion from the audience to Professor Dhar's talk, that many people did not try to believe that those what they call the junk DNA. I mean, what Professor Dhar, but this is not junk. This is probably a gold mine, or that's he's doing. So I also, when I was also doing my lab, I mean, but I also I have seen that how this Professor Dhar, from when other people thinks is negative, and how it makes is the positive in a few years, and specifically from India, where the resources is not that great, what Professor Dhar actually worked in the Riken or the Singapore. But still, what Professor Dhar inspired us, even from India, you can do some great work. Thank you, Professor Dhar. So you're always an inspiration to the students, as well as to the junior faculty like me. Oh, now I am not junior anymore. Thank you very much, Professor Dhar. So I'll just directly go to my talk, and okay, so one moment, please. I have. Can you see the screen?
Yes, it's perfect, sir.
Okay, thank you very much. So, so what I'm going to talk about is about the how do you do the computing with the synthetic genetic circuits, and that is one of the engineer. I mean, as Professor Dhar said, there's a you can define synthetic biology from a different perspective, but this is one of the perspective where the engineers the way they started the synthetic biology. So when I was in the end of my high school, that time, this is a big news, and it's in 1997 when IBM actually developed a computer. It's named Deep Blue, and it plays chess with Garry Kasparov, and you can see what is the result, right? The Garry Kasparov was one of the greatest chess players of the all time. And this is a big news throughout the world, and you can see that basically that a computer it actually defeated Garry Kasparov. So this is a computer. When you talk about, it is nothing but a computation. This is there's a lot of things inside. There's a lot of circuits inside. So in terms of most of these cases, this is the digital. This is the Boolean algebra they are working, and those circuits which is doing the computation, it actually defeated another level of the computation. There is the human computation through the brains, right? So we know there's sometimes we call the brain is one of the greatest known computer in the world, probably the universe. And so, so this is something computer versus computer, but definitely human brain is more than a computer. Human brain is not just for playing chess, but we can do everything. Today's meeting, when we are talking, whatever we're listening, whatever them is all come from this human brain. So human brain has the two things. One is definitely the computation and the intelligence, and another is really the consciousness. So we don't know anything about the consciousness, right? So we cannot talk at this moment what the consciousness is, but what we know that something about the intelligence, that intelligence is producing in the human brain by the computation of several, I mean, there's the billions of the neurons which are connecting together, and there is a 10 to the power 15 connections altogether on an average in a human brain. So from 1997 to 2021, we have actually crossed a big path in terms of understanding the human brain, and as well as to understanding the computation or developing something we call artificial intelligence. And this artificial intelligence still, it is not a match to the human brain, and we don't know when it will be. But there's a project, it's called the Blue Brain Project. It's the project from the Swiss Federal Institute and with the collaborations with IBM. And what they're trying to develop, and with an unprecedented scale, they want to simulate the human brain for each of these 10 to the power 15 connections between them. Okay, so that is the whole idea. But there is no guarantee that when you actually try to simulate that 10 to the power 15 different connectomes similar to the human brain, that is going to give you some great level of the intelligence or not. So there is so definitely, but people are trying to do that. However, a brain is not just a computation. If you see that when you try to simulate that 10 to the power 10 to the power 15 connections of the human brain through your computer or any other device, means it takes the energy of this. It's 1 megaton. So 1 megaton energy comes from a mini hydropower. So you need a mini hydropower to run a single human brain if you try to simulate that human brain in a computer, a supercomputer, or whatever you try to call it. However, biologically, a human brain takes only 20 words, right? It's nothing compared to this 1 megaton. So that's the way of the billions of years of our evolution, how developed the biology, not only to develop, there's a great level of the computation like a brain, but it also shows that how to use the energy, right? So that's so much operations are happening, like 10 to the power 15 operations or connections are happening. It just takes a 20 words. So that's something great thing about the human brain. So we all know in one way or another that human brain actually do compute, right? Now the question is, when you think about the evolution, is that computations only in the human brain? If you go to the very low level organism like a bacteria, can you see any computation there? So I just like to tell you something about, this is a old movie called Ghazni, and you can see that Amir Khan was the hero in this movie, and the character he played, Amir Khan forgets everything, and he wanted to take some revenge. That's why he wrote everything in his body, so whenever he forgets, so that he can just read and try to take that revenge. So there is a movie scene in that whole movie where Amir Khan was actually chasing a bad guy. So he was chasing, and suddenly there was a truck was coming from the other side. That truck actually hit that bad guy, and bad guy died there. And exactly at that time, Amir Khan forgot everything, and he looked so perplexed. So what this movie sequence actually told us, or to actually taught us, is that if you want to do a work, like if you want to do your immediate future work, you must remember your immediate past. If you cannot remember, then you cannot do that work. For example, when he was chasing that person, so he remembered the chasing, and then he was chasing. But at that moment, when he forgets everything, he could not remember what he did in his immediate past, so he cannot do in the future. Now, if you think about the bacteria, so something we all, it doesn't matter what is your background is, but we all learned a term called chemotaxis in our high school. In this chemotaxis, what happens, then there is a chemical attractant like a food particles, and then this bacteria can actually sense that and actually go towards that chemical attractant. Now, you think about an experiment when you have a beaker, and in this beaker, at one end, you actually put a sucrose crystal. Okay, and this sucrose crystal is actually diffusing from the other end to the other end. Now, on the other end, you place some bacteria like E. coli. Now, this E. coli can sense that. So now you can understand there would be a gradient of this sucrose. So this bacteria can sense that, and it starts swimming towards the highest concentration or the source of the food, right? So it will actually swim towards that. Now, what happens in the water? Water does not always stand still. There is a lot of turbulence. So this turbulence can actually hit that bacteria and actually put it in the other way around. So if this bacteria's direction actually changed, now bacteria stops. Bacteria don't swim, because if bacteria swims in the other direction, because just the water molecule actually reversed it, then it will go to the other side. So it cannot take the food and it cannot survive. So bacteria stops. So when another water molecule actually hits the bacteria and it again its direction actually changed towards the food, then again bacteria start swimming towards the food. So what this tells us, this is exactly like this Amir Khan's Ghazni moment. Because if a bacteria has to decide either I have to go forward or I have to stop, then it has to remember the concentration between two time points. Because if you want to go forward, then you have to remember the concentration of the previous time point. And if your concentration in the future time point is high, then you are actually going towards swimming. If it is low from the previous time point, it stops. So bacteria doesn't have the brain, but it remembers it. So it has a memory. So how this memory is coming? This memory is coming from the molecular interactions inside the bacteria. So that molecular interactions is creating or is computing to generate the memory, right? So this is, now if you really look about that, what level of the computation a simple bacteria can do, now I can give you this example. So at the left-hand side in my slide, you can see this is the network of the computers of the whole world which are connected to the internet. This is a 2009 picture, so definitely 2021 this complexity has increased, but this is just a 2009 picture available. And the right hand side, you can see this is a simplified representation of a biomolecular network of a bacteria. Right? Just looking at it, you can see there's the level of the complexity of the level of the connections are similar. And on the top of that, this bacterial biomolecular network, the interactions are nonlinear. There's a lot of feedbacks and there is a lot of the noise. So it is the computations this kind of network is doing totally is extremely complicated, and there is no mathematics to actually understand or depict that the whole level of the computation. So this computation, even a simple bacteria, what is happening inside its cell is extremely, extremely complex. Now, exactly at this point, synthetic biology came, and synthetic biology has a different kind of way or different kind of the philosophy to work towards it. And what they found the synthetic biology, because a set of engineers actually found that if you think about a computer, this computer starts with you can see the physical layers, right? And the physical layers are connected in such a way, it generates some logic gates, and these logic gates are arranged to develop a lot of integrated circuits and device, which you can call modules. Those modules you connect it together and make the computer. several computers connected to make the whole computer network connections. Now, similar kind of the hierarchy you can find in the biological cells. So you have the protein and genes, they're connected to each other to develop some biochemical reactions. Those biochemical reactions, many biochemical reactions together make a pathway, and these pathways are generating a cell. Those cells are developing tissues and cultures and the whole organism probably. Now, unfortunately, what are the actual rules from the protein of the genes to biochemical reactions to pathways is very difficult to understand from a biological point of view, because there are so many things, as I showed that the network is something like that. So what if we take some of the rules from this computer engineering? So for example, if you're going from physical layer to the gates, so you have some rules, or you're going to gates to the modules, you have some rules. So how, if we actually arrange this protein and genes in such a way that it produce some gates through the biochemical reactions, and then how these gates are connected to make a module, give a function. So if we connect these biochemical networks following exactly the same rules to make a pathway, then we can generate some of the new cellular functions, or we can control the cells in some predictive way, as the computer engineers do for the computer. So that is the whole philosophy of that, how you are using the computer engineering principle in the biological engineering in terms of the synthetic biology, where we're talking about this design based. And now what people have, when this philosophy came to the synthetic biology, or the starting of the synthetic biology, what people have developed? So people have developed a lot of different kinds of the logic gates, starting from the NOR gate, OR gate, AND gates, whatever the gates actually people used in the computer circuits. The only difference is in when you think about the electronic circuit, your inputs and outputs is the flow of the electrons. Either it is there or it is not there, 1 and 0. So similarly, in the biology, we're not taking the electrons, but we're talking about the biomolecules. So there could be a chemical inputs, either its presence is 1, its absence is 0, and it is going to produce some protein or some biomolecules, and we take this as an output, and it's you can tell that is presence and absence as that 0 and 1. So this way you can develop all kind of the toolbox. The whole problem is that okay, you can do that, it's great, but who cares? What are you going to do with it, right? And exactly where we should come at this moment, that when we can tell that why this actually the engineering principle is very important to the synthetic biology or in the biological engineering. So now, so let's talk about something, that how do a computer add numbers, right? So all of the computers, what we are using, it has something called arithmetic logic unit, and this arithmetic logic unit has something called adder. Okay, so adder is a circuit, something like that. It's truth table. And if we can generate this adder, connect a lot of adders, it can actually add many things. Similarly, for subtraction, there is something called the subtractor, and they're put together in a device. It's called arithmetic logic unit of the computer. Now, how we are going to develop similar kind of the adder, okay, in a biological cell. If you want to do, and exactly it has been, sorry. So what it has been done here, you can see in 2018, the people actually developed a adder, right, in a biological adder in the mammalian cell. And you can see there's so many gates, right? Now, the question is that if I tell you that you have to make a biological function like this, without this, if I don't give you this circuit, just tell that this is the 3 chemical inputs and you have a 2 protein outputs or something, and we have to make this by arranging the genes and proteins. In reality, making this kind of things is impossible. You cannot come up with any thinking that you can make an adder or a subtractor or like this kind of the truth table. So that is why the engineering comes, because engineering says that if you arrange some of the gates just like this, you can actually come exactly like an adder. So exactly what happened in this mammalian cell adder, you develop some biological gates the way I actually showed you before, and then you connect exactly in this way. And if you just connect in exactly this way, you don't have to think about this, how these molecules and the chemicals is working together, what is the feedback, how it is transcription factor is connecting to what, then how it is affecting the others. You don't have to think anything. You just have those biological cellular gates, connect them together, and then what you are getting is an adder. So this mammalian cell can actually add numbers. So that is why that engineering principle is required, because in engineering you have that modules and you just connect and you make the whole thing. So that's why you may be a PhD student, you may be a PhD or if great guys, but if you can see a very not that schoolgoers guy even can actually worked as a mechanic or even as a computer mechanic, because they actually have the modules, they connect and the whole thing. So that is the whole point of the engineering, you can do that. And that is why engineering is very important when you actually develop some new function, in a design-based function in the biology. So what is the applications of those things? So this is kind of one application you can think about. Developing similar kind of the principle, these people, they actually developed something called a classifier circuit. So this classifier circuit, so all different kind of the cancer cells have different level of microRNA signatures. Okay, different microRNA signature. For the HeLa cell has a different microRNA signatures than a normal cell. So these guys developed this classifier circuit, put in a HeLa cell, and when they put it in the HeLa cell, it matches. If it matches with this particular signature of this HeLa cell, then this HeLa cell actually they produce a protein and it actually turns into the apoptosis, the cell dies. But if you the same genetic circuit, if we put in the normal cell, this microRNA signature won't match. If microRNA signature doesn't match, this classifier circuit won't turn on and it won't produce any protein, and there is no effect in this cell. So you can see this how computation can be go towards the therapeutics. And here is several examples that how you can apply those kind of the computation, a very simple computation, but you can apply for a lot of fighting the disease in a programmed way. For example, here what they actually developed, they developed an engineered E. coli, okay, and you put inside your body because it's a kind of probiotics, and then if some pathogen actually goes into your body, then it produces some molecules. This engineered E. coli can sense that particular molecule and produce exactly a particular level of microbial, antimicrobial peptides, which kills only that particular pathogens. Not everything is not like antibiotic. So it is a very specific to that particular thing. So similarly, people try to develop something in mouse body. So it's called uric acid homeostasis. So you actually make a this kind of the programmed cell so that if there is a disbalance in the uric acid in the body, it can sense and it actually interact with the uric acid homeostasis cycle and balance this uric acids where it has to be. So this kind of things you can do. So there's a great application in the therapeutics through this programmed or programmed things. Now, if you go a little farther, we can see the computation is not just inside the cell, but throughout this body. So we all know that the human, as an adult human, how we actually grow, we started all of our life from a single cell, right? And that single cell actually converted, developed, and go through all the developmental cycle and come to an adult human. Now, and we have a different kind of the cells, and look, this of the kidney cells, we have the liver cells, we have the skin cells, all works differently. Yet each of them has the same genetic information, right? The same chromosome we do have and the same genetic information. So that the computation actually tells that how to grow, where to stop, which computations required for this cell, which computation this cell. And that's why, and it is, this computation is so robust, you never see that your liver cells at a certain point started making brain cells or your kidney cells starting making heart cells. If this is going to happen, then we all die. It is not a robust. So this kind of the robust computation is happening. So similar things is happening in the Drosophila or any other this kind of the animals. So the first time in 1952, this Alan Turing, who is also called the father of the computer science. So it is not only the father of the computer science, he developed a theory, something called the chemical basis of the morphogenesis. And he talked about something called reaction diffusion system. He said that so it is computing when it's developing, and this computation has the two things. It is chemical computation, and in chemical computation, some chemicals are reacting and some is diffusing. So if you actually just balance these two things, it actually forms a pattern. So exactly this. So this is a Drosophila when it's developing. When it's developing, it requires segmentation, and this segmentation, if we actually tag those genes, you can see there is a pattern of different kind of genes exactly. So this kind of the pattern formation is the very first thing to develop an organ or to develop the whole body of a human or the Drosophila or something. So this is 1952, right? And so if you, like in using the synthetic biology or any other means, if we can actually understand that computation and then engineer that computation something in vitro, what would be this application? So it has an enormous level of application is possible. So you know there's something called the tissue engineering, as Professor Dhar also mentioned about the tissue engineering. So today, when you talk about the tissue engineering, the whole idea that how in vitro you can grow an human organ. But unfortunately, you can grow the cells, but you cannot grow an organ because organ has a very complicated structure and complicated function. So people try to develop this kind of the three-dimensional form that people try to use something called material scaffold. So you make a material scaffold, put the cells in it, cells grow, and you can try to make an organ. And here is an example of such thing. And this is a 3D of a pig urinary bladder, the way I told that you make a material scaffold, put the cells and make this kind of 3D. But making heart with this material scaffold, nobody could do that. I mean, probably, I don't know with the material scaffold we can do that or not. However, if you look at our own life, when we are growing in our mother's womb, that time there is no material scaffold. So our genetic signature has some program in it, which actually builds somehow to make this pattern formation or whatever, which can make a heart. So if we, that kind of the pattern formation programming, if we can put it inside the cell, engineer the cells in vitro in such a way, then without the material, we can actually do that. So as synthetic biology says that if we actually engineer our own genetic circuitry from the cell, probably we can grow the cells like an heart. So that will actually a futuristic application and enormous application if we can actually really do that. So what synthetic biology have done till now? So here what you see, it's a pattern formation. So you engineered the pattern formation, but where? This is nothing but E. coli. This is a bacteria, and it is a bacterial lawn. And in this bacterial lawn, this bacteria produce either a red fluorescence protein or a green fluorescence protein. So these things are not actually plated like this. This is just a lawn. And but you engineered this bacteria in such a way, if you give a set of inducer, it can form the pattern the way you mathematically design it or computationally designed it. Okay, so this kind of the pattern formation is similar to that, though this is just two things, just one or two concentric cycles, but it looks something like this pattern. This is the first step of the pattern formation. Now, how it is computing? So now you see in this 5 picture, one of the picture is a computer simulation, just a computer simulation. Other 4 is the experiments. So you can see in the synthetic biology, we can engineer the computation in such a level that we cannot. Can you differentiate which one is the computation simulation and which one is the real experiment? You cannot. Even I forgot that which one is what. This is a 2005 paper. You can go to this Ron Weiss and Basu's paper and you can find that, right? So we can do that. So that's in 2005. And in 2015, a group of, another group of scientists, so another 10 years to take this from this 2 concentric cycle to maybe 5 or 6 concentric cycles similar to this, right? But so we are in synthetic biology, we are progressing that how complicated computing we can actually make a set of the bacteria, so and it's computing in such a way. So now it looks something like that. But again, it is 2-dimensional, whereas in Drosophila, this is a 3-dimensional. And synthetic biologists again went further and actually did a 3D patterning in mammalian cell. Now this mammalian cell, they are growing in a 3D pattern. At the same time, they have this pattern, this red and green things are not random. They're computationally, you engineered so that it's developed this kind of the pattern. So we are very slowly, we are going towards the where we can probably one day make an make an organ without any material scaffold and the proper functioning things. So if you can engineer those kind of computation properly in the cell. But it is not just, now if you think about its application, it is not necessarily it is just a therapeutic. It have a different kind of application. So which means, you know, the computation of the developmental biology, you can engineer it. So you think about, instead of building a house, you engineer the developmental biology of the trees in such a way that you can grow your own house. Okay. And it's a MIT efficient project, but nobody actually did that yet. But no idea that you have to do that. But what I'm trying to say, that if you can actually engineer the developmental program of the tree, you can actually program it such a way to develop a structure for you, a bridge or something like that, right? So that would be the great. So you can imagine that if the synthetic biology, when it in the full force in the next 50 years, probably you can do some marvel which cannot be done in conventional biotechnology or genetic engineering. Now I changed the gear a little bit, and I'll just come that what we are doing in our lab for last 6 years or so. So at SINP, so one of the goals of our lab, that we question that can we make bacteria artificially intelligent? If we can make a bacteria artificially intelligent, at what extent? So that is the whole idea. So you know this artificial intelligence, and behind this artificial intelligence, what you hear, there is something called artificial neural network. So what it is, so now, as I said, we do not know much about the brain, how it works. Okay, we know very little, but even if we take that very little knowledge and apply, we can actually do marvel. So in reality, like if you take a neuron, this neuron takes a lot of inputs from the environment, and these inputs are summed up in such a way, and this is processed and it produces some outputs. But if you really take that mathematics of this and you draw in your exercise book on a notebook, it looks something like that, which you can call artificial neuron, and it has x1 to xn different kind of inputs. Each of the inputs has a particular kind of the weight, W1. You don't have to understand the mathematics, I'm just telling. So all these inputs along its weight is first summed it off, and this summing of function goes through another nonlinear function and gives the output. This is exactly happening inside the neuron. Now, if these artificial neurons, you connect many artificial neurons together, gives you artificial neural network. And this is just by changing the W values, you can do a lot of different things. For example, think about the Facebook. Right? In the Facebook, now if you take a group photo, and in this Facebook, someone tells, Facebook will tell you that, do you want to tag your friend, Mr. X or Miss Y or someone? And how this Facebook is finding that face matching with the billions of the face which the Facebook server has, because billions of the people are the in the Facebook member. So the way it does, it have this artificial neural network, and when it gets a picture, it divides in the pixels, and then is changing the W of the artificial neural network made from your face, and then searching for it. You're just changing W, and suddenly it will match with the of your photographs and identify it as you. Okay, so this works in that way. So you can do a lot of different things with this artificial neural network. And in Facebook, it takes like 60,000 to 70,000 different kind of nodes. You can find that, but that's great. So we can do a lot of things just by knowing that neuron works this way, right? Just input, gives a mathematical function, gives output, and you can actually make something things. So mostly artificial intelligence, what you actually find, and that all you see is through the software, through the computer, like the Facebook, the server is there and you can do that. Now, but people know that through that you can, if you want to make a robot, so if you have to control from a server like a Facebook server, that is difficult. You want that brain, I mean the robot's brain only on this robot. So you need not the software, but the artificial neural network like an hardware, like a human brain. So people use neuromorphic chips. This is a material-based chip, similar kind of the artificial neural circuits are printed on it. And this hardware actually work as an artificial neural network. People shows that this can be done with the photonics. I don't know how, I'm not an expert at it. And people do also show that just using in vitro DNA computation, this kind of artificial neural network can be implemented in hardwares. So what my lab is asking this question: can you use a bacteria as an artificial neuron and connect them together to make an artificial neural network out of the bacteria? So that is the goal, where that's the goal. A short-term goal, recently what we find that throughout this, synthetic biology, if we generally takes that the starting point of the synthetic biology is the 2000, because the two important papers is published in the Nature from one from Jim Collins' group, the toggle switch, and another is oscillator. And throughout this 20 years, what synthetic biology use a computing system, what I already told you, that about making a lot of different logic gates and connect them together and make a device to develop a function. So what we think that artificial neural network is another computing system. So why not, instead of this kind of the gates, we can actually start making artificial neural network, the bacteria. So we're bringing a totally different computation system to make some synthetic biology function. So that is our short-term goal. And to do that, we took two important device, and this device is one of the devices called decoder, and another is the encoder. So decoder and encoder is something people always used in different kind of the communication device. So whenever you send a signal, and that signal is basically first it's encoded to a fewer bits, and then it goes through the air. And when the other end of this receiver actually receive it, it takes that encoded signal, decode it, it's the original signal, and this is a code. So the encoder and decoder is very important system for different kinds of communication system. So what we tried to develop, a lot of different kind of the function, including the encoder and so this is the decoder and this encoder, not using its electronic circuits, but using an artificial neural network. So we developed a set of rules where you do not need the actual electronic circuit principle to make those things, but we go to so those rules I am not explaining here, that's we don't have the time, but just believe me for this point that we are actually develop a set of rules where you do not need any electronic circuit diagram. We develop the way that how you can, from a truth table, you directly divide in such a way you come up with an NN structure. So when you come to the NN structure, it has a mathematics. So if you want to do in the bacteria, what you have to do through this molecular interaction, you have to abstract this mathematics inside the cell. So how we are going to do that? This is just an example here. So we have some rules, so that if this is a bacteroneuron 1, I mean, we have different kind of bacteroneurons to make a function, and this bacteroneuron function we develop through some interactions at the so we developed a lot of artificial promoters such that different transcription factors works in a specific way to match with this equations. And when we do that, we experiment with those things in the bacteria and found some input output relationship, and then we started changing that W value in the equation by some molecular engineering. So we changing the number of transcription factors, so changing the rate constants of different biological reactions, the interaction constant, and so on. And we are coming towards the what is the optimum behavior. So just to show you the optimum behavior is, for example, is the bacteroneuron 1. What you can see here, this is the computer simulations of this bacteroneuron 1, and this is the experiment. So you can see that these things match very well. So this is the thing, that when you develop this kind of bacteroneuron, you have to actually match them with the experiment and the simulation. When everything is done, we just develop a mixed culture of those kind of bacteroneurons. Okay, and this bacteroneuron you saw here, it is a decoder. It is an experimental result of the decoder. So in decoder, it has two different inputs. Okay, and based on the combinations of the inputs, it had 4 outputs, right? And if you see here, these 4 outputs like arranged diagonally, if you have F1 to F4 outputs. Now, when you actually do the same experiment with our bacteroneurons bacteria, you will see that exactly at the diagonally, based on the IPTG and ATC, 2 different chemical inputs, that it produces some fluorescence, 4 different fluorescence proteins, and it produces exactly like this diagonally. You can see everywhere it is like black. So this proves that we able to use this bacteroneuron idea and able to prove a device like the decoder. So we go is a little more complex device like the encoder. Exactly, we develop this kind of things, and you can see here that we develop this encoder and similar other things we actually develop. So another important aspect, when we actually doing that, we find something called reversible computing. Till now, the computing AND gate, NOT gate, or whatever thing, this is called irreversible computing. And this reversible computing is an important, this is the heart of something called quantum computing. Probably you have heard the term quantum computer. It could be use the quantum principle, and you can actually generate a very, very fast computer with this quantum computing. Now, the quantum computing use a kind of the gate, which is not that irreversible gate like AND or not, those kind of, but it's a reversible gates. For example, there is a reversible gate called the Feynman gate. So what is the reversible gate? It has a unique way to actually encodes that basically take the inputs, and it has a output, but is a unique. You can get the input from the output. If you have a, for example, here is 0 0 0 0 0 1 0 1. This 1 0 diagonally this gives 1 0, and 1 1 it gives 1 1. So if you have output, you can get what is the input is, which basically means this logical reversibility won't allow to actually lost any information. But if you think about an AND gate is only when A and B would be 1 1, then you will get a 1 at the output. Any other cases is 0. So from this output, you cannot get what is the inputs are. So it has the two possibility in the reversible computing. One is thermodynamic reversibility, one is logical reversibility. We cannot make thermodynamic reversibility in a cell, because the cell or biological system is definitely an irreversible system. But what we generated, that logical reversibility. So here we developed the Feynman gate, we developed the Fredkin gates using the same principle. This is the experimental results, and we have in between all this kind of characterization, mathematical simulations, and all those. So this is the experimental results, what we have done with this. Now, so when you do this kind of computing, what you can do, you can go probably much more than just producing some of the device. Here, we try to solve a maze problem. So maze is a very important problem, not only the human culture, but it's also a computational mathematical problem. It's probably, if you have seen the Harry Potter, you know that's the wizard maze, how Harry Potter was solving this kind of the maze. So you need a human brain to solve it. So if you give this kind of things to the robot to solve it, it is difficult for the robot because it has to use some algorithm and actually develop that. So what we developed, which is very recently published in the last week, that we develop a set of engineered bacteria which works as a computational algorithm in a mixed culture, and they solve a set of, it's not just a solve, but it solves a set of possible mates. For example, if this is a mate structure, you can see it start to destination. So just in this size mate, there's a 16 different possibilities, and in this 16 different possibility, there would be only 3 different solvable mates, and any other 13 are unsolvable mazes. So as a result, so what we developed this kind of set of the bacteria in such a way that this set of the bacteria can solve this, solve those mazes, where this maze problem is developed through the chemical space. So that's either, so I mean, okay, I cannot explain the whole thing, it will take a long time, but just to tell that, so if we have, just by using chemicals 0 and 1, we can generate a maze, and if you give this bacteria, it will solve by producing a fluorescence protein or not producing a fluorescence protein. This is again the circuits diagram for different cell types, but you don't have to go to the details. But here is the result. So there are 16 different mazes possible. See, this 3 has the solution, and when there is a solution, you can see the fluorescence protein as expressing, and through this expression, you can see what are the pathways. So here there are 2 different pathways. Here is only one path, here is only one path, but these 3 are only solvable, and any other places this is only black, which means they are unsolvable. So our things is not only solve the maze problem, but it is also saying that how many of them are unsolvable and how many are the solvable. If you want to calculate this mathematically, this is not a straightforward. So we can use this computational using these synthetic genetic circuits, and it can actually solves a lot of these things. So what could be the So now I go to the another example, what we have done recently. It is not published, it's in the review at this moment. So we developed this Feynman gate I talked about. So first we developed a single cell Feynman gate. So this is, you don't have to come with this. This is just a simulation experiment, and this is the microscopy result. So we developed the Feynman gate in the single cell. Then what we tried to want to do, we want to we transfer this information to bacteria, to mammalian cells, specifically cancer cell, to downregulate some of the genes, okay, and in a logical way similar as the Feynman gates. So to do that, first we developed a bacteria, and this bacteria engineered, is surface engineered such a way. You can see here that engineered E. coli, it attacks the HeLa cells. So these are the HeLa cells, and you see this kind of pin-like things, that is the bacteria attacking these cells. Whereas if you just co-culture the non-engineered E. coli and the HeLa cells and then wash it out, you won't see this kind of pins, or this kind of E. coli is attacking. So this is our engineered E. coli, which is attacking the HeLa cells, and it can be done. People actually done that. This is not our discovery. Okay, people actually doing it for the long time. Now, what we have done when we engineered this bacteria, with this engineer, we connect this Feynman gate. So in the Feynman gate, previously I showed the output was 2 different fluorescent genes. Now, instead of the fluorescent genes, we put 2 anti-shRNA. These anti-shRNA are generally people used as a cancer drug for different kind of directed therapy or pathway-based therapy. And then we apply those chemicals, okay, give the input in the bacteria, in a mixed culture of the bacteria and the HeLa cells, and you see that the 2 genes is basically on AKT1 and CTNNB1, sometimes called beta-catenin. These 2 genes are silenced exactly same way as in the Feynman's way. So you can logically control. So you're giving your inputs in the bacteria, you're getting output from the mammalian cell through this gene silencing. So you can actually do this information transfer, and you can do those kind of things exactly in the Feynman gate. So what could be the application, the future applications of all this? There's a term people developed, it's called cellular supremacy. So means there's a lot of application where you need a computer in a micro level, but you cannot bring a computer in the micron level, right? So how do you do that? So even when people talk about this kind of the micro robots is never been in a micron size. It is like a centimeter or a few centimeter size. The reason you cannot bring all the circuits, all the battery and everything in this way. So here, the bacteria or any other cell which is really in the micron level, which does not need battery because it can take the food from the environment. Now you can actually just give those kind of circuits. You can program those cells in such a way it will do all this computational work for you and at the micron level. So this is the cellular supremacy, because where you need a computer, you do not have the computer, but you can use the cells to do this kind of. Think about that, develop that like some therapeutics, like delivering the shRNA drug using, like if you need a Feynman get kind of the logical control through your bacteria, you can do these things. Another things what you can think about is those maze problem. I showed that those maze problem for maze, not the biological maze, but the actual maze problem people use for a long time for the data encryption. And it's called cryptography and the steganography. So we showed that we can solve the maze through the bacteria. Then we can actually start a new way to data encryption using engineered cells, because then just like looking, there won't be a hacker to actually hack just computationally that what is happening there, because you have a cell has their own way to do that. It is difficult to hack. So we believe that's another level of the applications of this. So with that, I don't want to give a conclusion, but what I want to give the way we should look at the synthetic biology. So the biology, what we're looking is, it is starting from your origin of life to today's human and the thing. The biology is something what is exists already in the world, but the synthetic biology is something what can exist. We really don't know how much things we can exist, but it is an enormous possibility. There's a limitless boundary. If we understand the computation in the biological system and engineer that in other system, then we can develop a very new biology which does not exist. One thing already Professor Dhar showed, that there is a what he called the junk DNA, or junkomics, which is not using the biology, but it shows that it has the biology thing. Now, when you connect this to all kinds of the computation along with those kind of things, we really don't know what we can do or what we cannot do. And with that, I'd like to thank my team. They're the students who are doing the real works. They're very hardworking, very smart, and my funding. And thank you very much for your attention. Thank you very much.
So thank you very much, sir. Very, very informative and very complex actually. So we try to figure out how we can understand. So you did a lot of very, very complex work. So I would like to invite Ritu to ask if anyone have any questions. Sir, can you stop sharing?
Okay, yeah, one moment, please.
Good evening, everyone. Sir, there is no question. Attendees, if you have any question, you can ask.
Okay, so I think we can proceed. So Pawan sir, what really message you want to tell us to do and keep synthetic biology in India? Because we are very, very late and still 21 years has been passed. Still, we are not in the phase where synthetic biology really started. And we know you start some level of the synthetic biology. So what are your key messages you want to tell us?
Well, there are many, but we don't have time today. Briefly, I would like to say that, first, we need support from the industry, because unless there are jobs generated by the industry in the area of synthetic biology, it will be difficult, because it will become 100% export-oriented unit. And that's what is happening. Good PhD students always go abroad in the area of synthetic biology mostly. But now we have been able to attract some of them. And as you can see, Sangram's work is fabulous innovation in this area. And we also need good students. Because what we have personally observed is that there is a lot of interest, but very little passion. People quickly give up and then they start complaining. We need people at the ground level. Actually, we need it at every level. We need faculty members to come together and launch some network projects. Sangram and I, we have been talking about this, unfortunately, because COVID, we could not go forward. But we need network collaborations. We need, if one university cannot run a synthetic biology program, let there be a group of universities offering a program like this for the masses. I think we need to come together and talk to each other often. We need an IGM equivalent desperately in the country. Department of Biotechnology needs a long-term vision to support it for at least 10 years. Well, if that is going to happen or not, we don't know, but these are some of the wish lists. Thank you.
Thank you very much, sir. So my same question goes to the Sangram sir on behalf of all the participants. In which level are you looking synthetic biology in India and abroad at your level? And what are the key messages you want to tell us?
See, I think Professor Dhar said the whole thing, but I just want to compare two things. So let's say where I'm working, Saha Institute of Nuclear Physics. So this is established by Professor Meghnad Saha, who is the one of the greatest scientists of the India who actually developed the thermoionization theory. But whole point is, like, at the very same time, when we look at the life of the Meghnad Saha, now I just want to mention 2 points. It is before our independence, and this is exactly 1 year. I mean, I think the year is 1943, or I couldn't remember properly. That time, people asked Niels Bohr that what is the possibility of making a nuclear bomb or nuclear energy, not the bomb, but getting energy from this. So what Niels Bohr was telling that time that it is not possible for the humankind because probably God does not want, because that level of the energy comes to the human being, because that time people are trying to do that. At the same year, Professor Meghnad Saha started a course, a specialization paper in the nuclear physics in the Calcutta University in the physics students. And why he started, so he's telling that one day this nuclear energy would be an important energy that the whole world will work on it. So, and when the India will get independent, we have to tap that energy. We need that technology for that. So we need to train the people from starting from now. So you can imagine that time the Hiroshima has nothing, nobody knew that we can get these things, but this guy is thinking. But it is not just the nuclear physics. In SINP, if you come, I invite you all, please come one day after these COVID things. You will see in our display, there is an electron microscope. That electron microscope is the world's second, Asia's first, and world's second or third, because there's a debate, is the second or the third microscope. That is Meghnad Saha sent some M.N. Dasgupta, who actually developed that electron microscope. And today's, whatever the treatment for the cholera, for the leishmania or everything, it's just because of this electron microscope, because all the ultra structure of all those Indian and these tropical parasites are actually, so there's a set of 22 Nature publication that time, so which actually publish the ultra structure of those things, those parasites, and those whatever the drug is coming out based on those kind of the structure. So you can imagine that just after the independence, that time, 1950s, 1960s, that time, so people thought something which is the same as the world, what because this is the word second or something. And today in 2021, for the next last 7-8 years, we are just discussing how world is going in the synthetic biology, what we are doing to do that. So this basically means somehow we are unable to understand that it is not just probably synthetic biology, but many other fields, that what they're going to do. So I think like that so this has to be started anyhow, but this can be started only when people get interested, people understand that what is the potential of the synthetic biology, and then it is the possible. So as what Professor Dhar said, that it is very important to have some of the industries, definitely, but another thing is that a human, if the students doesn't understand what synthetic biology, what it can be do, then who will think? Because the student will be next, after 10 years, they will be the faculty, they will be the industry CEOs, and they are going to do that. So it is important that to tell the people that this is synthetic biology, this can be done, and this can be done from India. I think that is the very important. So in here, I just want to tell at this moment during the COVID, if anyone is trying to develop any synthetic biology course anywhere, okay, I teach definitely in SINP, but that's just due to the PhD student, okay, but I'm very ready to teach the synthetic biology course and the part for any universities, if anybody actually do that. I think from my perspective, that could be the human source development is the one thing we can actually tap onto.
Yes, sir, it's very nice, sir. Actually, we all need the support. It's not only here, everywhere we need the support for the expert who really know about the area. So I think there is the question. Ritu ma'am, please, can you ask?
Yes, sir. Sir, there is question by one of attendees. What is the difference between synthetic biology and metabolic engineering in terms of advantages and disadvantages? And how one problem must be approached with synthetic biology and other by metabolic engineering?
I think this question even Vijay can respond to, but then I will attempt to answer this. Well, it's a very important question. It's a very interesting and important question, because even now people have their own views about what is synthetic biology. And a lot of people think that it is a combination of genetic engineering and metabolic engineering and bioinformatics and some part of systems biology, all kind of cocktail people are imagining. Please understand that metabolic engineering is a much more mature branch. It has been there for many decades. What it essentially means is that you control the flux of the metabolites in a particular direction to overproduce a certain component. Now, how you do that is by checking different components, increasing the turnover rate, and then ensuring that the flux remains in a certain direction. So this is all about permutation combination. Earlier, people used to think that if they increase, let's say there's a 10-step reaction in glycolysis, you increase the turnover at every step, you might get a bigger product or a larger numbers of molecule at the last step, which didn't happen. This is typically what people used in metabolic engineering. But it was all about transferring. Now that's one part is, of course, overproducing things. Second is that you transfer a certain metabolic branch from one organism to another organism. That's another way of looking at it. Please understand that in all of these was permutation and combinations which were attempted. Synthetic biology is not completely different from this, but it is different in the sense that we are talking of standards. We're talking of standard parts. We're not talking of a normal part. A normal part would be the one which can behave in a different way that you do not know. You don't know the behavior of these parts. But when you take synthetic biology parts, you exactly know how they are behaving, at least by the data sheet that you've gathered. Now, when you're combining that, you might do a metabolic engineering project, but the parts would be coming from a standard library. Whether they behave in the same way or not, that's a different story. Most likely they will not. So you will have to again work out some combinations there. So the key message here is that the key difference is not about metabolic engineering versus synthetic biology, but it's normal parts or natural parts versus standard parts. Or you can say natural components, let's say natural operon or natural pathways versus standardized pathways. So it's all about standardization of the components that you do in the beginning to assemble higher order structures with a predictive behavior. In my view, that is the key difference. However, in the synthetic biology community, people have also done a random mutagenesis and directed evolution. That has also been long DNA synthesis. That has been one of the key technologies used in synthetic biology, wherein you don't worry about 10 different genes, you just synthesize them and then plug them in and see what happens. And so what you do is you do a microarray-like environment wherein you have, let's say, 10,000 strains. I think Vijay has used those technologies. When you have accelerated genome engineering platforms, you have 10,000 strains, 5,000 strains, and you don't care what happens in each cell, you care about the output. So you're not doing a rational design, you are doing a random mutagenesis, directed evolution, and a high throughput environment so that you get your design end of the day, whichever way it comes up, you leave it at that. So eventually the differences are not major. The differences is in terms of building the standards. That is my opinion about it, but we can discuss.
Thank you, sir. Sir, there is another question, last question by a student of economics. Question is, how synthetic biology works for economy or will work for economy?
I think Murli should answer that tomorrow because he's the best person from industry.
So tomorrow, as a speaker, he will explain more. But I know you are a person for the policy and economics for the synthetic biology in India. So,
see, from the economic perspective or the ROI perspective, you look for the low-hanging fruits. What that means is, you do not go for highly complex designs. You look for those molecules that are novel, that have got some application, whether you are making a new enzyme or whether you are making a new therapeutic molecule or a new transcription factor or a new signaling molecule or whatever you make, as long as you are able to generate some ROI or the return of investment, it will help in the process. But it's not that straightforward, because when you do this theoretically in the lab, you are able to do that. However, when you are competing with the industry, that's a totally different scale. See, for example, the biofuel projects were, bioenergy projects were funded heavily by DBT. A huge amount of money has been spent last 10 years, but still we don't see that, market application of this. It's because there are many aspects, which is not, it is not like a digital switch, you switch it on and off. But many times you feel that it might work, but somehow, but one of the biggest stories has been that of artemisinin. Even there, people who have discovered, they had huge problems in maintaining that scalability, and then the industry had to come in. So I would say eventually it is the industry that will dictate terms. It is the market, how attractive their products in the market vis-a-vis what's available already. Are you beating them on the price? Are you beating them on the speed? Or are you coming up with brand new product? Or are you delivering the existing product at a much reduced price? There are many variables. So again, industry has to come up with the solutions and a good answer to this problem.
Thank you so much, sir. So all the participants, so we are very happy to have the speaker on the synthetic biology, where we start with the basic to the advanced complexity of the synthetic biology. So tomorrow onward, we have the same as Danny from International Center for Genetic Engineering and Biotechnology, New Delhi. So he will talk more on genome editing and the metabolic engineering. And we have the Murli, he is the CEO of the Evalva Biotech Studies company. And this is the first company they have R&D in India. So we will listen. Then we have the Guhan Jayaraman from IIT. So onward, we will go into the more product side and the market. I think we can proceed with the ODAC times for the Pawan sir and
Thank you, sir. I, Ritu Chaudhary, Assistant Professor at Indrashil University, on the behalf of our university, I convey my sincere thanks to both the speakers, Professor Pawan Kedar and Sangram Bagh. Thank you so much, sir, for accepting our invitation and sparing your valuable time for such a wonderful, amazing talk in the field of synthetic biology. It is really an honor to interact with you, sir. We all feel privileged to interact and hear you. And because most of our attendees are students of MSc and BSc, so you both started with a very conceptual part and concluded with the updated information. I am sure that our student will be benefited with this updated and conceptual information. Thank you so much, sir. And I would like to convey my thanks to Vijay Singh, sir, for providing such a platform for us so that we can interact with such renowned experts like Pawan Kedar sir and Sangram Bagh sir. And I also extend my thanks to Dr. Bharti Dave, Dean School of Science, and Dr. J.S. Yadav, our Provost, all the technical team, and all the attendees. Thank you so much, everyone.
Thank you so much, Ritu. So, Saswa sir, you can close the meeting. So we will meet tomorrow again with the more exciting, more informative about the synthetic biology, metabolic engineering. So thank you so much, sir. Once again, thank you, sir. Thank you, sir.
Thank you.
Saswa sir, you can close the meeting.
Okay, sir. Okay.
Okay.